Started life looking like that. The point is, Mummy and Daddy got together and had a special cuddle, and the end result was that you started as this large female egg with all these sperm cells, these male cells, trying to... We all started life like that. We may not want to think of Mummy and Daddy having a special cuddle, but they did. But that's what you started life as. Okay. But the point is that one cell has turned into the many millions of cells that make you up. My second question then is, how many stem cells in the bone marrow have to divide every second to maintain the number of red blood cells in the body? Is it 20?
No.
2,000? 200,000? 2 million or 2 billion? The answer is D, 2 million. And the formation, zoom in a bit, the formation of new cells is an ongoing process through all of life. So new cells are being formed all the time. Your red blood cells are being formed, other cells are being formed. New cells come about because they go through what's called the cell cycle, and that's what we're going to talk about today. So, with regard to the cell cycle, I'll put these words make up the PowerPoint presentation that I can't show, but we'll just go back to the words and talk about them. So I'll put the PowerPoint on there. But that ability to grow and reproduce is a fundamental property of living organisms, that all cells can grow and divide. That's the thing. So it doesn't matter whether we're talking about individual cells, bacteria, or we're talking about multicellular organisms. All those cells are growing and dividing. And during that cell growth, as we've spoken about bacteria, during cell growth, new material is made, that cells take up the nutrients available to them and convert it to things like protein, nucleic acids, carbohydrates, lipids. Cells grow. And so they get bigger. As they get bigger, the volume of the cell increases. Is this good or bad? Why is it bad? Yes, the problem is the surface area to volume ratio gets worse as the cells get bigger. And so the cells can't interact with their environment as well as if they can as if they're small. So there is a pressure for cells to be small, so they can interact better with their environment. But as the cells get bigger, they double in size, the plasma membrane expands to prevent the cell from bursting. But as we've said, that can't go on forever. Cells can't continue to enlarge indefinitely. As the cells grow larger, there's an decrease in the surface area to volume ratio. That gets worse and worse as the cells get bigger. So the cells can't get bigger and bigger and bigger because they don't interact as well with the environment. So cell growth must be accompanied by cell division, so that one cell gives rise to two new daughter cells. That's why you started life as one large female cell, which then has got, over your lifetime, many cells that are there. One cell is divided into two. And you have differentiated. Your cells have differentiated into different sorts of types of cells. So you've got nerve cells, heart cells, other muscle cells, skin cells, all that different sorts of cells that there are in the body have come about by the process of differentiation. We're not going to talk about that. We're going to talk about that cell cycle, the growth and the division of cells. So the cells that are produced, these cells that are made during growth and division, can add to the total population. You started as one cell, there's more and more cells to you, we can add cells, a growing child or a growing adult or whatever. And you've now reached a stage where your numbers of cells are about constant, so that the number of cells remain the same. You've got some cells growing, you've got some cells dying, but overall there's no net increase. Or we can replace cells that have been removed. As we've said, red blood cells are being broken down all the time. They need to be replenished. cells of your bone marrow. So maintaining the numbers of cells in an adult. So both those things are important. When cells grow, we'll just do this very quickly, when cells grow and divide, the newly formed daughter cells are normally genetic duplicates of the parent cell. So the cell at the end, genetically, is identical to the cell that started. That's not the case with sex cells, sperm cells, egg cells, because they are different in that they contain less DNA for various reasons, we'll make sure we know at the end. But normally all your cells are identical. The only difference between them in the different parts of the body is what genes are turned on, what sort of cells they have become. So all the genetic information must be duplicated and then distributed. So there's two things. There's making new genetic information and then making sure it gets to the right place. They're two separate things. Making new DNA is one thing, separating it into the daughter cells is another thing. So cells progress through the cell cycle. That's the point we want to get over there. So the cell cycle is the period from when a cell has been born. So we'll probably have a cell cycle along here. Here is birth of a cell, and here is division over the cell over here. So that's the cell cycle. One cell becomes two cells. The cells divide over here. So we'll express it along this front row probably as the cell cycle. So the cell cycle is this period from when the cell's been born here to when it divides. And it's a fundamental unit of time for a cell. But early, you were a cell biologist years ago, you wanted to see what was going on. In eukaryotic cells, your cells, down a microscope, the most interesting things that people could see was what was going on here at division. These processes of mitosis and the division of the cell itself. So it didn't, the cells were getting bigger over that time period, but that was about it. It was what was going on here at the end of the cell cycle is what people could see. So early cell biologists could actually, could see what was going on at division, but not what was really going on along here. This looked very boring along here. There's actually a lot of activity going on, but they could see what was going on at the other end. So right at the end, we get the division of the nucleus. So over here, about here in the cell cycle, we get the division of the nucleus, and then we get the division of the cell itself. Two separate things, the division of the nuclear material, the division of the rest of the cell, the cytoplasm and the organelles, etc. And so we get these two things, the division of the nucleus, or karyokinesis, is those events of mitosis, and then we've got the division of the cytoplasm itself, the events of cytokinesis, the separation of the cytoplasm, the rest of the cell. So we've got cell being born, looks very boring, but during that process the DNA is duplicated. Then we go to where people could see what was going on, mitosis, which is about the separation of the DNA. And then we get division of the cell itself, cytokinesis. So, there are all the different events that we need to think of. And so the events of the mitotic phase, what goes on at the end, where the DNA is separated. That's all it is separating the DNA. Say all, it's a complicated process, but that's what's going on. The DNA has been duplicated earlier on, it's being separated over there. So the events of mitosis over here are a small part of the cycle. Cells spend the majority of their time all along here, which is this phase between divisions, interphase. And if we look at when DNA is made, we know that DNA is made earlier on in the cell cycle. As we said, mitosis isn't about making DNA. Mitosis is about separating DNA into the daughter cells. The DNA is made earlier on over here, where it's replicated in a certain phase of the cycle. So this phase here of interphase, mitosis, just about separating DNA. And so there are various phases that we need to be aware of. Right at the end, well, it's a cycle, so it's difficult to know where to start, but we've put mitosis at the end, division, karyokinesis followed by cytokinesis, division of the nuclei, division of the rest of the cytoplasm. But then we go back here to when the cell starts the cell cycle, the G1 phase, the first gap, where there is a gap which really involves the preparation for the synthesis of DNA. So we've got the G1 phase right at the beginning, where the cells are preparing to make DNA in the S phase, S for DNA synthesis. Then we have another gap, the second gap, where we get preparation for division, the G2 phase. So we haven't drawn it as a line, but we've drawn it as what we've often seen before, the G1 phase, the S phase where DNA is made, the G2 phase, which is the preparation for mitosis, which is the separation of DNA and the division of the cell itself. So G1, S and G2 is the interphase, as we've said. We will refer to the total time taken for a cell to complete the cell cycle, and that's the cell cycle time, the doubling time, or the generation time. And in microbiology, we've often referred to the doubling time, how quickly the cells double. How long does it take for the cells to go through the cell cycle, from being born to when they divide? So the doubling time or generation time. We get variations in that generation time, of course. The S and M phases tend to be quite similar for different sorts of cells, but the length of the G1 phase is quite variable. So it's this first phase is the one that seems to alter in different sorts of cells. So a typical G1 phase lasts 8 to 10 hours. Some cells will spend only minutes in G1, while others spend weeks, months or years. And in fact they move to a different phase called the G0 phase. So during that G1 phase, a major decision is made as to whether or not the cell is going to divide again. Why? Why does a cell decide here that we're going to carry on with all this? It's a bit like a student getting up in the morning. They think, yeah, I'll be there. Bob's lecture, 11 o'clock, I'll be there. Right? So I've got to get up at 8. Right? Okay, I'll be there at 8. So they get out, shower, walk to the station, get a tube, train in, buses, so they have to get a tube. They get to Goode Street, walk around here, 10 to 11, had trouble on the trains and whatever. 10 to 11 arrives, walk through the door, dripping with sweat because they've run in for a lecture at 11 o'clock, and they go, oh, sorry, can't be bothered. Do you do that? No, you don't. You make a decision at 8 o'clock, Bob's lecture, shall I go? You make that decision very early on about the rest of your day, whether you're going to go or not. So it's the same with a cell going through the cell cycle. There's no point making new DNA if the cell is not going to divide. Because making new DNA requires a lot of resources, a lot of energy. There's no point going through the G1 phase, making all that DNA, going through the G2 phase, and then going, oh, sorry, there's no point doing that. Exactly the same with a student deciding to get up or not. So there's no point making new DNA if the cell isn't going to divide. So cells that are arrested in G1, we said earlier on, go into this phase called the G0 phase. So all this about G1, S, G2, mitosis, division, is fine for cells that are going to divide. Most of your cells go and have a rest. They just sit there, there's G1, you've got some cells that are zipping through there and going through to division, all this effort going through, dividing. A lot of cells are just sitting here watching. Right? They're not actively dividing. They're busy, they're doing all the things that cells do, but not, or very, either not or very rarely dividing. At times, we'll talk about that later, they come out and go, okay, we need to carry on and go on through the cell cycle. But for the majority of their time, they are sitting here, not really doing anything with regard to division. They're doing all the other things that cells do, but not doing the dividing. So, that G0 phase. In some cells, there's a similar kind of arrest also in G2, but again, why, I'll ask the question then, why is there more cells that sit here, over in G1, compared with sitting over there, coming out of G2?
Because you've got the DNA.
Yeah, because this DNA making DNA. There is no point making that DNA unless you're going to carry on and go through mitosis. So there's no point making all the DNA and then going and sitting down. But some cells do, but there's less point. So in general, G2 is shorter than G1. G2 is the phase where we get preparation for mitosis. For example, synthesizing parts of the microtubules that will form the spindle that's going to be necessary to separate the DNA into the daughter cells. So that G2 so G1 is a preparation for DNA synthesis. The G2 phase is a preparation for mitosis so that the cells can separate their DNA correctly. So different sorts of cells, if we look, the plant, human, yeast, mitosis, G1, S, G2, and the doubling time in hours. So if a plant cell is just dividing, it takes about 29 hours to double. Right? So if a cell's just been born as a result of division, it's going to be just over a day till they divide again. Same with humans, the length of the different phases, about 24 hours, if they are just continually dividing. Yeast cells is much less. This is in minutes, because as we know, yeast cells are much simpler. Yeast cells only take about 120 minutes to divide if they're going through all these phases. So remember that cells will go into these G0 phases to sit down and have a rest, but if you're just dividing, that's how long it takes. Whereas E. coli, and I haven't put it down as G1, S, G2, because the processes are different. It isn't quite like that. But it might be 30 minutes, 20 or 30 minutes, if it's going to divide at its maximum rate. So the doubling time of E. coli is much less than yeast. Yeast is much less than human cells or plant cells, for example, is the message that we want to get over there. As well as replicate the DNA, which as we know occurs in the S phase, S for what? Synthesis of the DNA. The cell has to double all the other things in the cell, all the other organelles. So how's that done? So it's all right talking about the DNA, which is what we'll concentrate on, but there's everything else in the cell that needs to be replicated as well. So, we need to remember things like ribosomes are made by the nucleolus of the cell. So the cell needs, at the end of the cell cycle, compared with the beginning, twice as much of everything. So we're talking about the doubling of DNA. We need to double the amount of ribosomes, double the amount of mitochondria, because at the end, the cell is going to divide, and we want to make sure that each cell gets an even distribution of the DNA, of the ribosomes, of the mitochondria. So, the ribosomes are made by the nucleolus, the cytoskeleton, we get polymerization of subunits there, membrane, rough ER, smooth ER, make proteins and lipids, mitochondria and chloroplasts, we get the growth and division of existing organelles, that we can see that the existing organelles, existing mitochondria, existing chloroplasts, divide in two. So we get the increase in number by the division of those existing organelles. So if there were 1 million mitochondria at the beginning, at the end of the cell cycle there will be 2 million, right? That have come about by the growth and the division of those existing organelles. And that's because, that, as we know, mitochondria contain their own DNA, they contain ribosomes, they contain mitochondrial RNA, they can make some of their own proteins because they have evolved from bacteria. So, we need to remember it's not just DNA that we're talking about these different organelles as well. But the frequency with which the cells divide can be different between different sorts of cells. So that some cells are dividing a lot, other cells not. So it depends on the function of the cell in the body. So for example, continuously dividing cells, or, virtually continuously, rapidly dividing tissue, like the stem cells of skin. Why? Why are stem cells of skin often dividing? Yeah, you do that and see the dandruff coming off. Because it needs to be replaced all the time. A lot of skin cells are being shed all the time. They need to be replaced. Cells of the gut. Well, it's a cold day. Some bread is mouldy in your kitchen. The kitchen is nice and warm and moist. You go to it and notice that there is mould all over your bread. Mould, as we know, produces toxins, things like that. What do you do? You go and give it the birds, don't you? Yeah, you go and throw it outside and get the birds to eat. Now, I've never understood this. This bread isn't fit for you to eat because you know that the bread contains mould that produces toxins. Why go out there and poison the birds? Why don't you just get a shotgun and shoot them? Why do it so slowly? But that bread that contains now, birds are stupid, of course, and eat it. You're not stupid. Well, you're less stupid because you've thrown it outside for the birds, but you've not ate it yourself. So you've avoided those toxins. But the birds, of course, that take in all those toxins, what you don't want is those toxins to have their effect on the gut, cells in the gut, and we want to get rid of them all the time so that they're being replenished. So there's no great build-up of all the problems that are caused by toxins and other nasty chemicals. So, stem cells of the gut are replenished all the time so that they don't build up all the mutations and all the problems that might come about by eating all the bread that you've thrown out there that's gone mouldy. And bone marrow. Yeah, the blood cells that you require as well, that those cells have got to divide in the bone marrow so that they replenish your blood cells that are being broken down and being replenished all the time. So we can see why those cells are continuously being divisions occurring. Most tissues are dividing infrequently. As we said, most tissues are sitting here. So fibroblasts, hepatocytes, etc. are sitting here in genome. Occasionally they come out and do their, oh God, not again, I've got to divide again, make all that DNA and whatever. But most cells are infrequently dividing, whereas nerve cells, muscle cells, this is one of the problems with if you break your spinal column and whatever and become paralysed, then you can't, it depends on the damage that's been done, but it's very difficult, impossible to get those cells to grow back in the correct way and form a new, in humans, a new spinal column so that your limbs work again. So neurons, muscle fibres, effectively never divide. Other cell types do divide under normal conditions, do not divide under normal conditions, but can be induced to do so. For example, lymphocytes, white blood cells. If you get an infection, your number of white blood cells will increase as a result of that infection. So when exposed to a foreign protein, they begin dividing as part of the immune response. So that's one thing. that we need to think of. That those cells, some cells can be induced to divide as a result of a signal, and we'll come back to that in a bit. And most of these variations in generation time are based on differences in G1. As we've said, cells that divide very slowly spend their time in this offshoot of G1, the G0 phase. They come out of that division process. Let's not talk about, we haven't got time to talk about the rapid cell cycle. Let's start talking about the control of the cell cycle. So the fact that those cells that we've just said can be induced to divide rapidly, for example, or can be sitting there, cell can be sitting here, and there is a signal for it to divide. An infection, for example, and your white blood cells, the numbers will increase. It can be more, what's the word? Sort of more important than that. Has anybody got a knife on them? Yes, no? No. That's a bit. Let's see if I can find something. Bring my knife. I was in a rush. Somebody must have a knife. Somebody sharp. Somebody sharp. What? Okay. So let's imagine this is a knife object. It probably still cause damage. If I put that in your eye, it will cause damage. We can visualise it causing damage. But I need a volunteer. What needs a volunteer? If we put the end on there so we don't make a mess. Here is our knife, right? Let's stab this person. Just hold it there so we can see. So it's been stabbed. Now there are two alternatives. She will either bleed to death, right? It's been stabbed in there, in the neck. Give me dying. I said dying, not dead. So I'm dying. Okay. Star of the show is here. So one alternative is she dies, she bleeds to death. Otherwise, the platelets in the blood will stem the flow, but then, if we don't, what will happen next is that if we did this and came back after Christmas, there would be a big scar there, right? That's going to show or not. Yeah. This is not from intravenous drug abuse. This is a gardening accident. But you can see the scar. Why did we go, ugh? The alternative to this was bleeding to death, or you formed scar tissue because the cells around that wound will then produce a factor that makes the cells around the wound to divide and heal there. Otherwise, you bleed to death. Right? So, we've all got scars probably. So, okay, if we did that, we'd know. We'll look at your scar after Christmas when you come back. But we can see that those scars are produced because of a platelet-derived growth factor that says to the cells around this wound, all these platelets go, you lot have got to divide. So here's a cell there, just sitting there like this. Suddenly, this platelet-derived growth factor comes in and says, you've got to divide, or she's going to bleed to death. Or so you can form a scar, so that the tissue is mended. We should do a little bit more, then we'll take a break. But there are many variations are possible in the overall length of the cell cycle and the relative lengths of the time spent in the different phases we've said. Some cells go into G0, G1 phase is variable, all these sorts of things. So the variability tells us that the cycle must be well regulated. that there must be a lot of control going on about these cells that are suddenly sitting here and then suddenly have to come out of G0 and start dividing, etc., or to go into G0. So there must be a great set of regulation going on, and it's a very important topic. Cancer cells manage to escape these normal cell cycle control mechanisms. So the first thing that we want to mention is we sort of touched on it already, is about internal control. So there's internal control and external control factors. But the control system that regulates progression through the cycle, as we've said, must occur so that everything occurs in the right order, for example. Like making new DNA. There's no point making a mitotic spindle over there, or all the bits that are occurring there, unless all the DNA has been doubled. Right? There's no point in making everything over here, because that's a lot of effort, if this hasn't occurred successfully, for example. So it needs to make sure that things work at the appropriate time and in the appropriate sequence. They go in the right order. And we need to make sure that each phase of the cycle has been completed before we go on to the next one. So here is a cell in G1 at the beginning. The cell is preparing for DNA synthesis, so it needs to make all the right enzymes, etc. If the enzymes aren't there, no point carrying on, right? So making sure that everything is completed, and we need to then go into the S phase. The DNA has been synthesized. We need to make sure it's all there. It's all been done. There's no point having one chromosome going, nah, I'm not bothering. You know, the cell isn't going to survive. So it needs to make sure that everything is done in the right order and is completed successfully before we go on to the G2 phase, which is about making the mitotic spindle so that we can separate the DNA. And it must also be able to respond to various external conditions. If this knife suddenly comes along, causes bleeding, causes platelet-derived growth factor to be released, causes cells to divide. So the cells have got to respond internally and respond externally as well. And there are a series of control points in the cycle known as checkpoints to do this. So it's just like a checkpoint in going from, oh, you're too young to remember the checkpoints between East and West Germany. Anyone remember the checkpoints of East and West Germany? Yeah, you're just showing your age. You can remember the checkpoints. But to go from one part of Germany to the other before the Berlin Wall came down, who's heard of the Berlin Wall? No, it's history. It's history. But it was very difficult to go between the two. There were checkpoints. I never went, but there were checkpoints that people were checked as they went out. They checked as they came back. And crossing the border was not a trivial thing. And so it's exactly the same with the cell cycle, that there are checkpoints before the cells go on to the next stage in the cell cycle. So these checkpoints are the important thing. And so the sorts of things that we might look for in these checkpoints, here is the cell cycle, here is the G1 phase, here is the S phase, the G2 phase, and mitosis. So let's start here. The cell's just been born. We go through the G1 phase. So there is a checkpoint down here. Is the cell big enough? Right? Can it, in a sense, can it survive? Is it big enough, or is it a scrawny cell that we want to get rid of? So down at the bottom, is the cell big enough? Is the environment favourable? If it isn't big enough, then we need to get cell growth occurring so that it gets to the right size. Is the environment favourable or not? If it's not a favourable environment to do all these things, then there's no point going on. There are other questions at the end, this G2 checkpoint, and there are lots of checkpoints, these are just examples, but is all the DNA replicated? One of the questions over there. Let's zoom in on some of the questions. Is all the... No, that's not zooming. Is all the DNA replicated? That question needs to be asked. Is the environment favourable? Again, is the cell big enough? There's a checkpoint there for that. Over here in mitosis, are all the chromosomes aligned on the spindle? Because remember, mitosis is about separating the DNA. If they're not all lined up correctly on the spindle to separate into the daughter cells, there's no point carrying on. There's no point separating, leaving one chromosome going, I'll be there in a minute, wait for me, I'll be there in a minute. No point doing like that. You need to make sure that everything is right before you separate the cells into the daughter cells or whatever. So those are the sorts of questions that are asked in the cell cycle, those control mechanisms. No, that's not the way I want to go. You'd think after many years I know which button to press, but you always get it wrong. The control of the cell cycle. Done that. And one of the people about control, some few years ago, 2003, one of the people who got the Nobel Prize for controlling the cell cycle, as the title called, was Sir Paul Nurse, some of you might have heard of, been on telly, he's now president of the Royal Society. But the university invited him to come and talk, to give a talk to people about controlling the cell cycle in 2003. I lectured on it. Actually, I met him years ago, but as I lectured on it, I invited out to dinner with him. I think we'll normally get more impressed when you go out for dinner with the Nobel Prize winner. So we'll do that one again. So I went out for dinner with this Nobel Prize winner. Wow. What actually happened was, went on to the talk, and the person organising said, do you want to go out for dinner, Bob? And because I didn't realise I was, going to do that. So I phoned home and said to the wife, don't cook dinner, dear. I'm going out for dinner with a Nobel Prize winner. She wasn't impressed. So we went for dinner, and the university, Paul Nurse is a dead normal bloke. He drives a motor. He spent his Nobel money, you get 900 million quid or whatever. He bought a new motorbike, right? So he drove around London on a motorbike. And the university decided to get him a large bouquet of flowers to thank him for this. Flowers on a motorbike don't go well. So I said to him, I know, because me and support, we were good mates by now. I said, I got the flowers, I know you don't want them. Do you mind if I help them take them over to the wife? So, oh yeah, no problem. Yeah, I don't talk about it. It was quite late on by this time. So I arrived home, with this large bouquet of flowers, rather worse for wear. Sort of couldn't find the key, rang the door. Wife comes down to the front door, and I said, "These are for you." And she said, "You haven't been for dinner with a Nobel Prize winner. You've been with that woman again." Is that true or false? Well, everything's true, apart from the last time. She didn't say. She just sort of went, "Oh God," So the last bit wasn't true. Everything else was true. Anyway, but what was actually quite interesting was, all right. There's Sir Paul Nurse. There you go. He's a dead normal bloke. No more foreigners. Paul Nurse really was the first time he searched for his father. He went after he was here. He went over to the United States to run the Rockefeller University in New York. And now he's come back and he's head of the Royal Society. But when he went over there, you've got to get a green card to go and work over there. Yeah. They wouldn't give it to him. Here he is, probably the most eminent geneticist in the world. He didn't know who his father was. The people who he thought, he didn't find out until he applied to go to the States, because when they looked at the records and whatever, the person who he thought were his parents were really his grandparents, and that his let me get it right, who he thought was his sister was really his mother, because she got pregnant at 16. And those days, that was bad news and whatever. So the grandparents brought him up as their own, and his elder sister was really his mother. Right? The most eminent geneticist in the world didn't know his own history. Right? So this is why human genetics can be a bit tricky. People lie, you know, because, oh yes, there's my father. 1 in 10 people, it isn't. You know, all these sorts of things come into it. But you don't know it's not your father. He didn't know who his father was. Okay, let's stop there. Let's take a break and then we'll finish off there. And I'll try and get the PowerPoint. So we just take 10 minutes. It's 12 o'clock. If we can be back just after 10 past, please. Cancer cells are about. That they die rather, they keep on growing, they've forgotten how to die. Let's move on and just say a little about, in the last quarter of an hour, if you've got 6 pairs, you're a fly. If you've got 11 pairs, you're a toad.
Okay.
If you've got 12 pairs, you're right. Now, I don't see what the logic is there. You know? Very, very similar number of chromosomes, very different. Toad and rice. 13 pairs is a frog, so you could see toad and frog, you know, being similar. If you've got 16 pairs, you're an alligator. 21, you're a monkey. If you've got also 21, you are wheat. If you've got 23, you're human. If you've got 24, you're a potato. I don't. You may see a logic in this, but I don't. Yeah, and the most complicated, okay, with 52. Right? Is a cock, is a goldfish. Right? The most complicated is a goldfish, which I don't know if you've got a goldfish at home, but they swim around looking stupid. They don't do much. Why that's got twice as many as you, or more than twice as many, I don't know. Anyway, we're talking about chromatin structure. Okay. So, after the DNA, so you've got 23 pairs of chromosomes, other cells have got other numbers, as we've made there. After the DNA molecule replicates during S phase, the chromosome consists of 2 joined sister chromatids, right? And so, the long molecules to start with, but we end up with a pair of sister chromatids that are joined at a specific small region, the centromere. So here is our pair of sister chromatids joined here at the centromere. So this side and this side are identical. Okay, that's the point that we're making there. So they've been doubled up in number. So each chromosome has made these pair of sister chromatids. And we need to think about, well, how are we going to separate those into the daughter cells? Because, right, I'll tell you what we're going to do now before you jump back, right? We're going to separate DNA back. Okay. What we're making on a larger scale, here's our 2 sister chromatids, and we'll put it there. We'll put it there because we're going to separate it. Here's our 2 sister chromatids joined together at the centre. A bit of sellotape there, right? So here we are, our sister chromatids. That we're going to separate it. You're going to grab hold of that side, and I'm going to grab hold of that side. And we'll do it on the long bit. You're going to pull that one, and I'm going to pull that one, and it's going to go
So that's what we're going to do. Very simple. The process of mitosis is all about separating DNA into the two daughter cells. You can put it down quietly so you don't frighten yourself. I'm going to make a loud noise. Oh, sorry. It's end of term, you get sort of very sort of short-tempered. So, we have our sister chromatids there, and we need to have some sort of way of separating them. It's very easy to do that, but we need, for example, what is this? It may appear a football to you. To me, it is a mitotic apparatus. I took this from my children. I said, no, they want it. I need this at work. No, don't take my football, Daddy. Please don't take my football. No, I'm having it, I said. Oh, get down. I'm taking it. So, now we can see what's going to, how this might work. That if we put our sister chromatid on here, and this pulls in this direction, and this pulls in this direction, we'll separate the two. And if we had another, we've got 23 pairs of these things, or we can nearly be a mosquito, and we can sort of line them up on there, and they all get pulled over in the different directions. This was years ago when I stole this. My children now. You want that football, Dad? You really want that football? They'd hit me back. Anyway, so in mitosis, which occurs in the majority of cells in the body, a single nucleus gives rise to two nuclei that are genetically identical. That's the important thing there. The process ensures the accurate distribution of these chromosomes to the daughter nuclei, and it is a continuous process that we divide up for convenience into different phases. And those different phases, of course, if I can find my diagram, are, first of all, that of prophase, right? So in prophase, the chromosomes, as we can see, consist of two sister chromatids. So there's prophase, prometaphase, metaphase, anaphase, and telophase. They run continuously into each other. They have certain characteristics that occur at different stages, but they run on into each other. A cell doesn't stop at the end of anaphase and go, look at me, we've done this or whatever. It's a continuous process. It's just that when we look at diagrams or whatever, we see something that is particular to that stage. So, in prophase, we see these sister chromatids. Remember that the DNA was doubled. The DNA was doubled here. That this set of DNA was not packaged up, but this occurred in the S phase. This doubling of the DNA occurred here. All this is about separating that DNA that's been replicated into the daughter cells. One goes in that direction, one goes in that direction. It's only about separating, that's all it is. So it's not about making DNA, it's about separating DNA. So, in prophase, we've got these sister chromatids becoming more visible. Prometaphase, we're starting to get the breakdown, or we've got the breakdown of the nuclear envelope. So it depends on the sort of cell, but the nuclear envelope may disappear. But then, in metaphase, over here, the chromatids line up on this metaphase plate. That's the important thing, because these are going to go off in this direction, these are going off in that direction. So, here is the metaphase plate in this cell down the centre, and so we see, let's do it like this. So here is our cell with these fibres going that way, those fibres going that way. Here is our this has got 1, 2, 3, 4. I've only got 1 there, right? This is taken that direction, that's taken off in that direction. I'm not going to separate these because I'll use this again. But in anaphase then, we're starting to get the separation of the DNA into the different daughter cells. So interphase, it's all wrapped up. Prophase, we're getting condensation. Prometaphase, just off the end, the nuclear envelope breaks down. Metaphase, they line up on the plate. The anaphase, they start to separate. And then we get telophase, where we're getting it not breakdown of the condensed form, and we're starting to get actual division of the cell itself, cytokinesis occurring. So prophase, prometaphase, metaphase, anaphase, telophase, and then division itself occurring. So as we've said, mitosis is that separation of the DNA. There's a single nucleus, 2 nuclei that are genetically identical, ensures accurate distribution of the multiple chromosomes, a continuous process that we divide up into those pictures for convenience, not because it stops between each one, it goes on. So that's mitosis. We need to, we'll stop after we've just spoken a little about meiosis, which occurs in gamete sex cells. And that's important. Now, what we're talking about here, the birds, the bees and the flowers. And as we know, bees take pollen from one flower to another. This is why this flower has a smile on her face, because the pollen that's been produced is either just about to arrive. No, it looks as if it's been taken away. Its bag is full of pollen, right? That pollen comes in various shapes and sizes. Why? Why is pollen, which is a sex cell, right? It's like sperm cells or egg cells or whatever. But why does pollen have these wonderful shapes? Different types of pollen. Because basically plants have sex with something a few miles away, right? When this pollen there, so there's a plant. Can you be a plant for me? Be a plant. Right. And here is another plant over that we need to have. We don't have sex here. I just want you to be clear where this is going. We're going to have sex. But if you're a plant, if you're a plant, it's a bit difficult, right? We need, and you need to control who you have sex with, because you don't have sex with him. You don't have sex with him, right? You want to have sex with me. You need to make sure that you have sex with me, not with him. How are you going to do it, right? When you're a long way off. If you're human, right, of course, it's much easier to, because you can control who you have sex with. Because it's the same. Would you like to have sex? The response is no. Right? It's very easy for humans to control who they have sex with. Right? But if you're a plant, that's much harder to do. And you do that. Thank you. You do that as a plant by controlling which pollen that fits exactly right to open or whatever happens in flowers, to get to the other bits that it needs to get to. So you can control it by which one do you like the best? The one that looks like a teddy bear. How do I look like a teddy bear? But okay, here's this teddy bear floating around. Floating around in the air, or carried by a bee, or whatever, and then to get. You wanted the teddy bear, you've got a teddy bear. But now you can decide. That's how plants decide, by making sure the fit is spot on with something of the same species, whereas humans do it by slapping your face or whatever. We'll stop by saying that in meiosis, the functions of sex cell formation, the functions of meiosis are to reduce the chromosome number from diploid to haploid. Because when those two cells fuse together, then we need to get back to the diploid number. So sperm cells and egg cells, or pollen cells and the cells in flowers are both haploid, because when they fuse together, they become diploid. We need to ensure that each of the haploid products has a complete set of chromosomes. That there's no good getting only half of them there, and we need to promote genetic diversity among the products. So, two sorts of haploid meiotic cells. We've seen before the large egg cell, sperm cells there as well. That's meiosis, but we can get mitosis. I don't know if anybody's got one of these or ever had one of these, but they yell loudly. They're just as a result of those meiotic cells dividing together. Mitotic divisions result in this. If you've never had one, you don't know how loud a child can cry in the middle of the night. I think we need to stop there, because I think there's a little class on in here. I had all that on PowerPoint. I'll put PowerPoint on. Thank you. I'll put that on Blackboard for anybody who wants to look at it there. Your presentations this week or next week, let me know. If you can't do it this week, those on the Wednesday, let me know. Please send me an email.